Hi, my name is David Vega, and I'm a senior pre-vet in Ag and Natural Resources double major with a minor in equine science. This summer, I'm working in the Biddle lab on genomic sequencing of single protozoa cells from equine fecal material.
Protozoans make up the majority of the gut biomass due to their large size. However, they are still an understudied population within the equine microbiome. Morphological identification is the current gold standard for protozoa profiling, but molecular methods are an attractive alternative that produce reproducible results with quicker and easier methods. The goal of this study is to single-sort unsequenced protozoa species and correlate 18S gene sequences with morphological identification. This project will expand the 18S database and further the understanding of equine gut protozoal communities. Our single sorting methods require a fresh fecal sample which is then suspended in 1x PBS solution. Say goodbye to the horses! Once we're back in the lab, the fecal PBS slurry is filtered through a mesh strainer and a stomacher bag. This solution is then slowly transferred through a series of increasingly smaller filters to attain different size fractions of protozoa cells. Each filter is cleaned off with PBS solution and transferred to a vial. The sample is now ready for single sorting. Equal parts of the sample and PBS solution are transferred to a 6-well plate, which is then looked at under an inverted microscope. Using the TubeView software, a computer is easily connected to the microscope for ease of single sorting and capturing pictures and videos. Here I'm using a pipette set at 1 microliter to pick up the single protozoa cell. Using TubeView, I captured the moment where I picked up the unsequenced protozoa Blephariconus bembroki. This sample will now be transferred to a PCR tube, where it will undergo a series of lysing and freeze-thaw steps. The sample is then put into the thermocycler for PCR amplification and will later undergo gel electrophoresis to see if the correct band size is achieved. This is an example of 2 samples that correlate with the correct band size in relation to the latter. Once the correct bands have been identified, we can now send the sample for genetic sequencing of the full 18S region. Within our current sample population, 33% of these species haven't been sequenced. Therefore, the data collected in this study will fill the gap in this knowledge and improve future studies wanting to use molecular techniques to identify equine gut protozoans.